A small animal lowers itself into the grass at the edge of an open field, somewhere in the dark hours before the first gray of morning finds the horizon. Its ears settle, its legs fold beneath it. The muscles along its back, which have been holding it upright and ready through the long evening hours, release by small degrees into the ground. The eyes close, or close most of the way, the lids dropping to a thin and trembling line. This is sleep beginning, not the sleep of a protected creature in a sheltered place, but sleep in the open, under the wide and unguarded sky, with the field stretching out on every side and the dark offering no particular safety. The animal is a rabbit. Its heartbeat slows only a little. Its breathing deepens by the smallest measurable amount. And somewhere inside the biology of this moment, a threshold is rising, the threshold of arousal, the invisible line between the world getting in and the world being kept out, rising just enough to matter, and not an increment further. The grass is cold where the dew has begun to settle on it. The air carries the smell of soil and the faint mineral trace of standing water somewhere near. Sound reaches the field from a long distance in the dark, a low movement of wind, a thing shifting in the tree line, and the rabbit does not stir. It is asleep, in the only way a rabbit in an open field can afford to be asleep, which is lightly and briefly and always at the edge of waking. Within a few minutes, it will rouse again, not because anything has alarmed it, but because its sleep is built in pieces, short, shallow episodes that flicker in and out across the night like a lamp with a poor connection. Each bout of sleep lasts only seconds to a few minutes before the animal surfaces again into a thin wakefulness, looks, listens, smells the air, finds no immediate cause for alarm, and settles once more. By the time the field begins to lighten, the rabbit will have slept in dozens of these fragments, and the total will add up to something, though it will never have been still for long enough to be truly unguarded at any single moment. The way a rabbit sleeps is not an accident. It is not a failure of sleep, or a lesser version of sleep. It is sleep that has been pressed into this exact shape by millions of generations of selection pressure, the unforgiving arithmetic of predation across geological time, grinding the architecture of rest into something that the body can sustain and the open field can tolerate simultaneously. What a rabbit does in the grass at night is what sleep looks like when survival costs have been factored into every second of it. That shaping, the way an animal's sleep bends and fragments under the weight of danger, is one of the first and most important things biology has to say about why sleep exists at all. Because if sleep could simply be avoided, if it were merely a convenience or a habit or a surplus, then the prey animals of the world would have found their way around it long before now. They have not. They sleep in pieces, and at great cost, and without anything that looks like choice in the matter, which means that something inside sleep is necessary enough to be worth even this. A rabbit in a field at night is carrying a question inside it that biology has not fully answered. The question is an old one. It is older than rabbits, older than fields, older than the particular arrangement of continents and climates that produce this darkness and this dew. The question is simply this: why sleep at all? It seems at first like a question with an obvious answer. Rest is necessary. The body needs time to recover from the demands of the day. Everyone who has ever been very tired knows that sleep is not optional, that the body will eventually take it by force if it is not given freely. But obvious answers in biology are worth pressing on, because the body does many things that look like rest from the outside and are nothing of the kind, and sleep is stranger and more specific than the word rest suggests. Consider the energy, One of the most straightforward arguments for why sleep evolved is that it saves energy, that the body, by reducing its activity during the dark or the cold or the quiet, conserves the resources that would otherwise be spent on movement and alertness, and sleep does save energy. The metabolic rate of a sleeping mammal is measurably lower than that of a mammal doing nothing at all, simply lying awake in the dark. Lower, but only by something in the range of 15 to 20%. A modest saving, not trivial, but not the kind of number that immediately justifies the cost. And the cost is real. It is not metaphorical or theoretical. The sleeping animal cannot run. It cannot detect a threat as quickly or respond to one as decisively. Its awareness of the world is reduced in a way that could, at any moment, be the difference between another day of life and something much shorter. The 15 to 20% metabolic saving has to be weighed against that, against the persistent and measurable risk that something in the dark will close the distance while the animal's arousal threshold is raised, and its muscles are slow, and the world is slightly further away than it should be. 15 to 20% is not enough, not by itself. If energy conservation were the whole story, evolution would almost certainly have found a cheaper way to achieve it: quiet wakefulness, reduced movement, a shallow torpor that did not raise the arousal threshold and slow the response to threat. But no lineage has done this. Every animal that has been carefully studied rests in a way that meets the specific behavioral criteria that researchers now use to identify sleep: a period of quiescence, a raised arousal threshold, the ability to return quickly to full wakefulness when disturbed, and the drive to recover lost sleep with extra sleep when it has been prevented. These four markers: quiescence, raised threshold, rapid reversibility, homeostatic rebound, appear across the animal kingdom with a consistency that suggests they are not four separate adaptations, but one. The mammal pressing its metabolic rate down by 15 to 20% in the sealed chamber of the laboratory is also doing something that quiet wakefulness does not do. The cleaning runs faster when the brain is in slow wave sleep. The immune system shifts its emphasis. The memory of the day's events is being processed and stored by circuits that are not available for that purpose when the eyes are open and the world is pressing in. Something is happening in sleep that cannot happen any other way, in a body that has not figured out how to do it while standing up and watching the tree line. The energy story is the thin edge of a much larger thing, and the larger thing is still, in the most honest scientific sense, not fully understood. Somewhere in the mid-1980s, in a laboratory at the University of Chicago, an experimental apparatus called the Disc over Water Platform was being used to determine what happens when sleep is taken away entirely. The apparatus was designed by Alan Reschaffen and his colleagues to answer a question that sounds almost too simple. What does the body do if it is never allowed to sleep? The rats in the experiment were monitored continuously, and every time sleep began, as measured by the electrical signature of the brain shifting toward its sleeping pattern, the disc they stood on began to rotate, carrying them toward the water, waking them by the necessity of staying dry. The rats were never hurt in any direct way. They were fed freely, and they ate. They were housed in controlled conditions. Every physiological variable that could be measured was measured, and the record of what happened to them across the following weeks is one of the most uncomfortable data sets in the history of sleep science. Not because it is dramatic, because it is so quietly relentless. The animals lost condition, their fur grew ragged, their weight fell even as their food intake rose, the body burning more and more fuel without being able to make use of it properly, as though the thermostat had been broken and the furnace was running at full power while the house grew colder. Their body temperature became harder to regulate. The systems that normally managed infection began to fail, and microorganisms that a healthy rat's immune response would have contained without difficulty began to spread. Within 2 to 3 weeks, the animals died. They died not from any single dramatic failure, but from a slow systemic unraveling, thermoregulatory collapse, septicemia, a body that had been pushed past some threshold it could not return from. And the specific cause of the unraveling was not the absence of food, or of water, or of safety, or of warmth. The cause was the absence of sleep. All those other things had been provided. Only sleep had been withheld, and the animal had died of it, in the same slow, inevitable way that a person dies of thirst or starvation, as though sleep were a nutrient as essential as water, as necessary as food, as non-negotiable as air. This is the center of the paradox, and it is worth resting in for a moment without trying to resolve it. Sleep is dangerous. Sleep in an open field at night is a risk that the rabbit accepts dozens of times in a single darkness. Each brief episode, a small gamble against the probability that nothing with teeth is close enough to matter. And yet the only alternative to this risk, across geological time, across every lineage and every ecosystem in which animals have been studied carefully, appears to be death. Not inconvenience, not impairment. Death on a timescale of weeks, even when every other biological need is met. Something inside sleep is so necessary that the body cannot survive without it, even though the body must be made vulnerable to obtain it. This is the paradox that has sat at the center of sleep science for as long as sleep science has existed, and it has not been resolved, and the attempt to resolve it has produced some of the most careful and intricate science of the last half-century. The biologist David Sampson and others working in the evolutionary biology of sleep have framed the argument in a particular way that is worth considering slowly. If sleep could be eliminated, if some lineage of animals, anywhere in the history of life, had stumbled onto a way to obtain whatever sleep provides without the vulnerability of unconsciousness, then the selection pressure to do so would have been immense. Predation is not a mild inconvenience. It is the primary force shaping the behavior and morphology of prey species across every ecosystem on Earth. The animals that survived were the ones that avoided being eaten, and being eaten while asleep is a real and constant possibility that every prey species on the planet contends with every night. Over hundreds of millions of years, selection pressure this strong tends to produce results. It produced the speed of the gazelle, the camouflage of the octopus, the armor of the tortoise, the poison of the dart frog. It produced the fragmented sleep of the rabbit and the unihemispheric sleep of animals that need to keep one half of the brain watching even while the other half rests. It has compressed sleep durations and shifted sleep timing and altered sleep depth in species after species. But across all of this, across every adaptation and modification and architectural compression, it has not, in any lineage yet confirmed, eliminated sleep. No fully sleepless animal has ever been found. Every major animal phylum that has been studied with the behavioral rigor that the 4 criteria of sleep require has yielded animals that sleep, or that enter states so functionally analogous to sleep that the distinction is a matter of semantics rather than biology. The absence of a sleepless lineage is not a small thing. It is a negative result that has accumulated across hundreds of millions of years and thousands of species, and it carries the same weight in evolutionary biology as a positive finding. The pressure existed. The benefit would have been enormous. The time was more than sufficient. And still, no animal lineage found a way to remain permanently awake. What this means is that whatever sleep does, whatever it is providing that the body cannot obtain any other way, is so fundamental, so non negotiable, so deeply woven into the biology of animal life, that evolution, with all of its creative power and all of the time in the world, has not found a substitute. This is remarkable. It is perhaps the most striking fact about sleep, and the one that makes the question of why sleep evolved not merely an interesting scientific puzzle, but something closer to a first principle of animal biology. The jellyfish is worth considering in this context. The upside-down jellyfish, a marine animal that rests on the seafloor rather than drifting through open water, has been observed in recent years doing something that looks, when examined carefully, unmistakably like sleep. Its pulsation rate, the rhythmic contraction of its body that draws water across its feeding surfaces, slows measurably during the night hours. If it is disturbed during this slow period, its response is sluggish, its return to activity delayed. And if it has been disturbed repeatedly through the night, it pulses more slowly in the hours that follow, as though repaying a debt. Quiescence, raised arousal threshold, homeostatic rebound. 3 of the 4 behavioral markers in an animal with no brain at all, only a diffuse net of neurons spread across its body, like a loose mesh. The jellyfish lineage diverged from the lineage that would eventually produce vertebrates and insects and human beings more than 500 million years ago. 500 million years of separate evolutionary history, and both lines arrived at a state that works, behaviorally, like sleep. Either sleep evolved once, in some ancestor so ancient and so simple that it had not yet developed a centralized nervous system and has been carried forward in every lineage since, or it evolved independently, more than once, in animals so different from one another that the only explanation for the convergence is that the underlying need is universal enough to produce the same solution every time. Both possibilities lead to the same place, which is the recognition that sleep is not a late addition to animal life, not a luxury developed by complex organisms with complex brains, but something that arose at the very beginning of animal existence and has never been successfully abandoned in all the time since. Picture this as a slow numerical descent through geological time. 500 million years ago, the animal lineages were just beginning to diversify. 400 million years ago, fish were swimming in the first seas, in forms recognizable as fish. For example, 300 million years ago, the ancestors of mammals were moving across land. 200 million years ago, the first true mammals were small and mostly nocturnal, pressing their tiny bodies into whatever warmth the night allowed. 100 million years ago, the world was warmer and louder and crowded with forms of life that no longer exist. Through every one of these intervals, in every lineage that would survive them, sleep was present, not as a curiosity, not as a vestigial habit carried forward by inertia, as a biological necessity, taking different shapes in different bodies, compressed and fragmented, and modified and elaborated by every imaginable selective pressure, but never eliminated. The rabbit in the field at night is the near end of this long thread. Its fragmented sleep is what the thread looks like in a small prey animal that has spent many millions of years being shaped by the proximity of things that want to eat it. The brevity of each bout, the shallowness of each episode, the ease with which it surfaces back into wakefulness. All of this is the signature of selection pressure made visible in sleep architecture. But underneath the fragmentation, the thing itself is still present. the debt accumulates, the rebound happens, the arousal threshold rises. Sleep, in whatever compressed and cautious form the rabbit's ecology has required it to take, is still unmistakably sleep. The risk that sleep imposes on a small prey animal sleeping in an open field is not a small thing. It is not a minor tax on survival. Every moment of raised arousal threshold in an exposed animal is a moment in which the probability of being taken has increased by some real and measurable amount. And natural selection is very good at finding and eliminating behaviors whose costs outweigh their benefits. The fact that sleep has persisted, has been retained in every prey species ever studied, under every kind of predation pressure, across every ecosystem and every geological interval in which animals have left their record, is the strongest possible evidence that the cost of sleep is lower than the cost of its absence. Something that the body does during sleep is so necessary that the animal unable to obtain it dies faster than the animal that obtains it in an exposed field with its arousal threshold raised, and the dark all around it. The rabbit that sleeps in brief, fragmented bouts in the open is not making the best of a bad situation. It is making the only choice available, because the alternative, no sleep, is not a choice that any confirmed lineage has ever survived for long. The metabolic chamber tells one small part of this story. The oxygen consumption curve drops as a sleeping mammal breathes in the sealed and measured air, and the drop is real, and the number is 15 to 20%, and it is not enough by itself. It is not enough to justify the raised arousal threshold. It is not enough to justify the vulnerability. It is not enough to explain why no lineage, across all of the time that has passed since the first animals began to sleep, has ever found its way around the necessity. The chamber number is a beginning. It is the smallest and most legible piece of a larger accounting that biology has been trying to complete for more than a century, and has not yet completed, and continues to press toward with every experiment and every observation and every carefully constructed apparatus designed to remove sleep from an animal and watch what fails. Rechtschaffen's rats ate more food as they deteriorated. This is one of the quiet details in the record that resists easy explanation and sits in the mind with unusual weight. The body deprived of sleep did not reduce its demands on the outside world, it increased them. It burned more fuel than usual, as though trying to compensate for something it could not identify or replace. And the compensation did not work. The thermoregulatory system failed anyway. The immune system failed anyway. The body temperature fell. The animals continued to eat. Whatever sleep was providing, it was not simply fuel. The food was available and being consumed, and the absence of sleep was still fatal, which means that sleep provides something that food cannot replace, some form of processing or clearing or recalibration or restoration that the waking body cannot perform on itself, no matter how many resources it is given. The hungry sleeping body does this work during rest. The sleepless body, given all the food it asks for, cannot do it at all. The rats are the proof of this, written in the most unambiguous biological terms available. And yet what the work is, the specific, mechanistic account of what sleep does that cannot be done while awake, remains a matter of active scientific investigation, with multiple serious answers proposed and real evidence for each, and no single answer that has been shown to be complete. This is not a failure of science. It is an accurate description of where the science is, and it is more honest and more interesting than any simplified answer would be. What can be said, standing at this beginning, is that sleep persists because not sleeping is worse. Whatever sleep costs in vulnerability, in exposed minutes in open fields with the arousal threshold raised and the world slightly further away than it should be, the cost of its absence is higher. This is the conclusion that 500 million years of evolutionary history appears to support. No animal lineage that needed to sleep has successfully dispensed with the need. The pressure to do so has been as strong as any force that evolution exerts on behavior, and the result has been fragmentation, compression, architectural modification in every direction, but not elimination. The thread runs back through all of animal life and does not break. It runs through the jellyfish resting on the seafloor with its pulsations slowing in the dark. It runs through the first fish that ever rested in the slow current at the river's bottom. It runs through the small nocturnal, insectivorous ancestor of all mammals, pressing itself into the root hollow while the great reptiles moved overhead. It runs forward through every lineage into the rabbit in the field, into the bat folded in the cave ceiling, into the elephant standing asleep in the dusty heat of the afternoon, into every sleeping body that has ever been measured and observed and studied, somewhere in the deep past of animal life, before there were brains as science now understands them, before there were spinal cords or skulls or anything recognizable as a nervous system in the modern sense. Some first animal settled into a state of reduced activity with a raised threshold of arousal and the capacity to rebound when the opportunity for rest was taken away. It settled into this state because the biology that drove it toward that state was the biology that survived. And from that moment, however far back in geological time it lies, the thread of sleep has run forward, without interruption, into every animal alive today. The absence of a single confirmed sleepless lineage across all of this time, against all of this pressure, is not a small silence in the fossil record. It is a vast and telling quiet, the sound of an evolutionary outcome so consistent that it carries the weight of a natural law. Not a law written in equations, but a law written in the behavior of every animal that has ever been watched carefully in the dark, in the flickering sleep of the rabbit, in the slowing pulse of the jellyfish, in the slow descent of every sleeping body into a state that something in its biology absolutely requires, that evolution has never found a way around, and that science, after more than a century of careful looking, has not yet fully explained. The field is dark. The rabbit has settled again into another brief episode of shallow rest. The dew is cold on the grass. The arousal threshold is raised just enough and not a fraction more. The mystery runs all the way down to the biology of this moment, and all the way back through time, and in every direction outward to every sleeping animal alive on the planet tonight. It is one of the most ordinary things in the natural world. It is one of the least understood. Across 500 million years, through every lineage and every extinction and every remade world, through ice ages and warm, shallow seas, and the slow drift of the continents across the face of the planet, no animal has managed to stop sleeping. The geological record of this persistence is written not in rock but in behavior, in the raised arousal thresholds of jellyfish and insects and birds and mammals, in the homeostatic rebounds of creatures separated by more evolutionary time than the mind can comfortably hold, in the fragmented sleep of every small prey animal that has ever survived the night by resting in the only way the open world permitted it to rest. The question of why has no single clean answer. It is a question that reaches into the deepest parts of biology and finds in every corner it enters real evidence and partial explanations and the quiet persistence of things not yet known. What the question has, instead of a single answer, is the accumulated weight of 500 million years of consistent behavior in every branch of the animal kingdom, a weight so great that it can only mean the thing is necessary, even if the full account of why it is necessary continues. In laboratories and field stations and sealed metabolic chambers around the world to be carefully and patiently worked out. The numbers descend 500 million years, 400 million, 300 million, 200, 100, each interval containing the same quiet fact, present in every lineage that survived it, sleep in every form it has taken, persisting, the world changing around it, the lineages branching and diverging, and some of them ending, and sleep continuing through all of it, in the body of every animal that made it through. The rabbit settles into the grass, the dew falls, the dark holds, for now, whatever moves within it. And in the small sleeping body in the field, the biology of something ancient and unresolved and absolutely necessary goes quietly on. In a small laboratory in Lyon, sometime in the middle of the last century, a researcher sat watching a sleeping cat. The room was quiet. The cat lay still. Its breathing was slow and even, its limbs arranged in the loose way of a body that has surrendered to rest. By every measure visible to the eye, the animal was deeply, completely asleep. And then the researcher looked at the paper scrolling out of the electroencephalograph beside the bed, and what he saw there did not match what he was seeing with his eyes at all. The trace was fast. It was irregular. It moved in the quick, low, jagged pattern of a brain that was wide awake and busy, a brain sorting through the world and reacting to it, a brain far from anything that could be called rest. The cat had not moved. Its muscles were, if anything, more limp than before. But its brain, the brain of this still, quiet, sleeping animal, was running at something close to full speed, producing electrical weather that looked almost indistinguishable from the weather of waking. Michel Jouvet had been expecting slow waves. Everyone expected slow waves in a sleeping brain. What he found instead was something that had no name yet, no category, no precedent in the literature of sleep. He called it paradoxical sleep. The paradox was precise and genuine. The body was more deeply asleep, less responsive to the world outside than at any other point in the night, while the brain was doing something that looked, electrically, almost exactly like thinking. That observation, made in a French laboratory in a sleeping cat in the early 1950s, opened a door that researchers have been walking through ever since, and the room behind it has turned out to be far larger than anyone expected. The sleeping brain is not quiet. That much is now established beyond any serious doubt. What it is doing during those hours of stillness, what the purpose of its elaborate activity might be, why the body has arranged matters so that the most dangerous period of each day is also a period of intense and precisely organized biological work. These questions are the ones that remain, patient and unresolved, at the center of sleep science. The EEG trace of a sleeping brain across a full night looks like weather. During the first descent into sleep, the waves grow slow and wide, rolling in long intervals across the paper, each crest separated from the last by a second or more. This is the sleep that most people picture when they think of sleep, the deep, slow, quiet kind. The breathing settles, the heart rate drops, the muscles release. Down in the hippocampus and the cortex and the thalamus, the electrical activity organizes itself into something almost like a slow, synchronized tide. These are the slow oscillations of deep NREM sleep, and they are among the most precisely engineered signals the brain produces. Set beside them, The trace of REM sleep looks like a different instrument entirely. Where slow-wave sleep writes in broad, sweeping curves, REM sleep writes in narrow, rapid scratches, fast and low amplitude and irregular, a handwriting that belongs to a wakeful brain rather than a sleeping one. The two landscapes could not look more different. They are separated on most nights by a transition of only minutes, the brain shifting between them with a smoothness that makes the contrast even stranger. Both are sleep, both are as far from waking as the night gets, and yet they look nothing alike, and the biological work happening in each one is as different as the electrical patterns that mark them. What happens in slow-wave sleep, in those long rolling oscillations, begins in a small and ancient structure tucked at the center of the brain. The thalamus sits near the geometric middle of the brain, roughly an inch and a half below the top of the skull, a paired structure about the size of two walnuts placed together. In waking life, it acts as a relay station, routing sensory signals from the body and the outside world up into the cortex, where they become perception. In sleep, the thalamus does something different. Its relay nuclei begin to fire in a slower rhythm, coordinating with a thin shell of inhibitory neurons called the reticular nucleus that wraps around them like a net. The reticular nucleus quiets the relay neurons in rhythmic pulses. The relay neurons, after each pulse, rebound, firing a brief burst before quieting again. This rebound, enabled by a specific type of calcium channel built into the neuron's membrane, is what generates the characteristic waveform of NREM sleep, the sleep spindle, a brief waxing and waning of synchronized activity lasting a second or two, visible on the EEG as a sudden tight oscillation riding on the background slow wave. The slow waves themselves are generated in the cortex, in the large pyramidal neurons of the neocortex, whose long axons reach down through the brain and whose activity sets the rhythm for much of what the sleeping brain does. During NREM, these neurons cycle through two alternating states, an up state, during which they fire rapidly and intensely, and a down state, during which they fall almost completely silent. The up state is loud. The down state is near total electrical quiet, a silence in the cortex that has no real equivalent in the waking brain, a moment when most of the neurons of the outer brain simply stop. These up and down cycles, repeating roughly once per second across the hours of deep sleep, are what appear on the EEG as the slow waves that gave this stage of sleep its name. Between the thalamus and the cortex, in this rhythmic conversation of spindles and slow waves, something else fires. In the hippocampus, a curved seahorse-shaped structure buried beneath the cortex on each side of the brain, brief bursts of synchronized activity appear in precise coordination with the thalamic spindles and the cortical slow waves. These bursts are called sharp wave ripples. They are fast. Each one oscillates at between 80 and 120 cycles per second, far faster than the slow waves they accompany, and each one lasts for only 50 to 100 milliseconds, a tenth of a second or less. They appear at a specific moment in the cortical cycle, always during the up state, always coordinated with the thalamic spindles, arriving in a three-part rhythm that plays out across the dark hours of NREM sleep with the regularity of a clock. Georgi Bousaki, a neuroscientist who has spent decades mapping the electrical architecture of the sleeping hippocampus, characterized these ripples and their timing with a precision that made clear they were not random noise. They were a signal. Their temporal coordination with the thalamic spindles and the cortical slow oscillations was too exact, too consistent, too specifically organized to be coincidence. Something was being communicated in that three-part rhythm. Some transfer of information was moving through those precisely timed bursts, up from the hippocampus toward the cortex, during the hours when the body lay still and the eyes were closed and the world outside registered only as something to drift across without waking. The best current picture of what this three-part rhythm is doing has to do with memory, and it holds that the sleeping brain is not resting from the work of learning, but continuing it by other means. During the day, when a person moves through the world and accumulates experience, the hippocampus acts as a first receiver. New information, the layout of a room, the sequence of a task, the face of a person just met, arrives in the hippocampus and is held there in a labile, temporary form, encoded in the pattern of connections between hippocampal neurons that fire together in response to the new experience. The hippocampus is fast and flexible. It can bind new information quickly, hold many things at once, and retrieve them easily in the short term. But it is not a permanent store. The cortex is the permanent store, and transferring information from the hippocampus to the cortex, integrating new memories into the vast distributed network of existing knowledge, is slow, and it appears to happen primarily during sleep. The sharp wave ripples are thought to be central to this transfer. When the hippocampus fires a sharp wave ripple, it reactivates, in compressed form, the neural sequence that was active during some waking experience. The place cells that fired as a rat moved through a maze, the feature detectors that encoded a human face, the motor sequence that was being practiced during the day. That compressed reactivation arrives in the cortex during the upstate of the slow oscillation, when cortical neurons are already active and primed to receive input. The thalamic spindle, arriving at the same moment, appears to help stabilize the cortical activity that results. The pattern is written a little more deeply each time the cycle repeats. Over the course of a night, with hundreds or thousands of these coordinated bursts, the transfer from hippocampus to cortex appears to be completed, the temporary holding in the fast, flexible hippocampal store released, and the memory distributed into the slower but more permanent architecture of the cortex. This is a picture of what the sleeping brain might be doing, not a settled account of what it is certainly doing. The evidence for hippocampal reactivation during sleep is strong. The precise mechanism of the transfer and the extent to which this process fully explains the memory benefits of sleep remain active areas of research rather than closed questions. What is clear is that the three-part rhythm of slow oscillation, sleep spindle, and sharp wave ripple is real, is precisely organized, and is associated with measurable improvements in memory for experiences that preceded sleep. The sleeping brain is doing something with the day's experience. What exactly, and how, and to what end, these questions have partial answers and many open edges. The cleaning that happens in the sleeping brain was not known at all until fairly recently. The brain is an extraordinarily metabolically active organ. Though it accounts for roughly 2% of the body's weight, it consumes close to a fifth of the body's energy during waking hours, running its billions of neurons through millions of firing cycles each second, maintaining ion gradients across membranes, synthesizing neurotransmitters, sustaining the continuous electrical activity that wakefulness requires. All of that activity produces waste. Metabolic byproducts accumulate in the spaces between neurons, in the narrow gaps and channels of the brain's tissue that are collectively called the interstitial space. And among these byproducts are proteins that, if allowed to build up, appear to damage neural tissue over long periods of time. Amyloid beta is one of these proteins. It is a small peptide, a fragment of a larger protein, that neurons produce as a normal part of their metabolism. In a healthy brain, It is cleared as quickly as it is produced. In brains where clearance fails, for reasons that are still not fully understood, it accumulates into clumps and plaques that disrupt the connections between neurons. It is one of the defining features of Alzheimer's disease, and the failure to clear it efficiently is thought to be central to the disease's development. And what Macon Nedergaard and her group at the University of Rochester discovered in 2013 was that this clearance runs fastest not during waking, but during sleep, and by a specific and previously unknown mechanism. The brain, unlike most organs, lacks a conventional lymphatic system to drain waste from its tissues. What it has instead is a network of channels that run along the blood vessels threading through brain tissue, channels formed by the long processes of astrocytes, the brain's most abundant supporting cells, lined with a water channel protein called aquaporin 4. Cerebrospinal fluid, the clear liquid that fills the spaces around and within the brain, can move through these channels in a directed flow, sweeping through the interstitial space and carrying metabolic waste with it, eventually draining the tissue of byproducts that would otherwise accumulate. This system Nedergaard's group named the glymphatic system, combining the role of glial cells with the lymphatic analogy. What they found was that during slow-wave sleep, the interstitial space, the gaps between neurons, expands by roughly 60% compared to its waking volume. The cells themselves, particularly the astrocytes, appear to shrink slightly, opening up the channels between them. Into these widened spaces, cerebrospinal fluid flows more freely, and the rate of metabolic waste clearance increases substantially. The flow is not passive. It is driven by the slow oscillations of neural activity, by the rhythmic changes in neural firing that characterize NREM sleep, and by the pulsation of blood vessels whose walls move in coordination with the slow waves. The sleeping brain is, among other things, a brain that is washing itself, moving fluid through its own tissue in a regular rhythmic tide, carrying with it the molecular residue of a day's worth of thinking. The full implications of this cleaning process are still being mapped. In humans, a single night of sleep deprivation produces measurable increases in amyloid beta concentration in the brain, detectable with the imaging techniques now available. Whether this represents a meaningful long-term risk, and what the relationship is between chronic sleep disruption and the accumulation of proteins associated with neurodegenerative disease, are questions that active research is working through. What Nedergaard's finding established is that the sleeping brain is not metabolically idle. It is running a different metabolic program, one that appears to prioritize clearance over activity, maintenance over processing, the slow work of keeping the tissue clean over the fast work of responding to the world. The brain does not simply go offline when sleep begins. It reorganizes. Among the most studied networks in the waking brain is a distributed set of regions that activates when a person is not focused on any particular external task. When the mind wanders, when attention turns inward, when the brain is, in a loose sense, at rest. This network includes the medial prefrontal cortex, which sits behind the forehead and is involved in self-referential processing, the posterior cingulate cortex, a region near the back of the brain's midline associated with memory and self-awareness, and the angular gyrus, a region at the junction of several lobes involved in integrating information from different sensory modalities. Together, these and the regions connected to them are called the default mode network, a name that reflects their tendency to activate during the absence of directed external tasks. In sleep, this network does not simply shut down. It undergoes a stage-dependent reorganization that mirrors, in a loose way, the stages of sleep themselves. During the early descent into NREM sleep, connectivity across the network weakens. The medial prefrontal cortex and posterior cingulate begin to decouple from each other, their coordinated activity falling apart as sleep deepens. The self-referential processing that this network supports, the constant low-level narrative that waking consciousness maintains about the self and its situation, dims and quiets. During REM sleep, partial re-engagement appears. Some of the connectivity that was lost in NREM returns, and the pattern of activation during REM resembles, in certain respects, the pattern of waking, the same regions active, but their activity less coordinated, less stable, and organized according to different rules than those of either wakefulness or slow-wave sleep. What this reorganization represents in terms of what is actually happening to conscious experience during sleep is not easy to say. The default mode network's activity during waking is associated with self-awareness, with the continuity of personal identity across time, with the kind of background processing that keeps a person oriented in the world. During sleep, those processes are suspended or transformed in ways that no one has fully characterized. The network's behavior during sleep offers a window, not yet a clear one, into what happens to self-referential processing when the ordinary experience of being a self is interrupted each night by hours of something else entirely. The electrical activity of the sleeping brain is in the end a kind of map without a fully legible legend. The terrain is visible. The slow waves, the spindles, the sharp wave ripples, the reorganizing default mode network, the glymphatic tide. These features can be measured, their coordinates fixed, their timing recorded, what each feature means, what it accomplishes, how it relates to the others, and to the life of the waking person who lies unconscious while it all proceeds. These are questions with partial answers, growing answers, answers that change as new instruments allow closer measurement, and as the biology becomes better understood. The map is real, the full legend is not yet written. Deep in the base of the brain, in a region called the basal forebrain, a slow chemical accumulation happens across every waking hour. As neurons fire during wakefulness, they consume energy in the form of adenosine triphosphate, the cell's universal energy currency, a molecule that releases energy when one of its phosphate bonds is broken. The broken bond leaves behind adenosine, a smaller molecule, which diffuses out of neurons and accumulates in the spaces between them. Adenosine is produced wherever neurons are active. Across the waking brain, wherever firing is happening, adenosine is building. But the basal forebrain, which contains clusters of neurons important for maintaining wakefulness, is particularly sensitive to its accumulation, and it is here that the chemical's sleep-promoting effects are most pronounced. Adenosine acts on two receptor types in the brain, designated A1 and A2A. Both are present in the basal forebrain and surrounding regions. When adenosine binds to these receptors, it suppresses the activity of neurons that promote wakefulness and simultaneously reduces the inhibition of neurons in a region called the ventrolateral preoptic area, a small cluster in the hypothalamus that, when active, promotes sleep. The result of adenosine's accumulation is a gradual tipping of a molecular scale. The wake-promoting neurons quiet. The sleep-promoting neurons, freed from inhibition, become more active. The brain begins its slow withdrawal from the alert, responsive state of waking, and the weight of accumulated adenosine pulls it toward sleep. This mechanism was given a formal structure by Alexander Borbély, a Swiss sleep researcher whose two-process model of sleep regulation, proposed in the 1980s, described the interaction between a homeostatic sleep drive, the pressure that builds with each hour of wakefulness and dissipates during sleep, and the circadian rhythm, the roughly 24-hour oscillation in alertness and sleepiness that runs independently of how long a person has been awake. Adenosine is the molecular substrate of the homeostatic process, the chemical accumulation that represents the brain's running debt of needed sleep, the tally of all the hours of firing and thinking and perceiving that have accumulated since the last time the debt was cleared. What clears it is sleep itself. As NREM slow-wave sleep proceeds, adenosine is broken down and removed from the basal forebrain. The molecular pressure diminishes. The wake-promoting neurons, no longer suppressed, begin to recover their tone. The sleep-promoting neurons of the ventrolateral preoptic area, their activity now less needed, gradually reduce their output. Over the course of a night, the chemical balance shifts back from the heavy, almost irresistible pull of high adenosine toward the lighter, more alert baseline that makes waking possible again. The brain wakes not because it has been told to, not because something external has imposed consciousness upon it, but because the chemical debt accumulated during the day has been paid, and the neurons that sustain wakefulness can fire freely again. Caffeine works, and this is where that mechanism lives. Caffeine does not produce energy. It does not generate wakefulness in any active sense. It binds to adenosine receptors, to the A1 and A2A receptors in the basal forebrain and elsewhere, and blocks them, preventing adenosine from delivering its sleep-promoting signal without reducing the amount of adenosine present. The debt accumulates regardless. The caffeine wears off. The adenosine that has been building, blocked from its receptors for hours, now floods them all at once, and the weight of the full accumulated debt arrives in a rapid cascade that in some people produces an abrupt and powerful wave of sleepiness. The debt was always there. Caffeine only delayed the accounting. The depth of this adenosine system, the precision of its molecular machinery, the way it coordinates with the circadian clock and with the electrical architecture of sleep, all of this speaks to something about sleep that resists easy summary. Sleep is not a single thing. It is a collection of biological processes running in parallel, each with its own molecular basis, each doing something that the waking brain either cannot do at all or cannot do as efficiently. The cleaning of the tissue by cerebrospinal fluid, the transfer of memories from hippocampus to cortex through the coordinated firing of slow oscillations, spindles, and sharp wave ripples, the clearing of the adenosine debt, and the restoration of the molecular conditions that make sustained wakefulness possible, the reorganization of neural networks in ways that are still being mapped. These processes are simultaneous. They are interleaved. They are in the hours of sleep, happening all at once in the same brain, in the same dark, quiet body, lying still. And still, across all of this, something that feels like a question persists, patient and large, and genuinely unanswered. The processes are real. The molecular machinery is real. What remains unresolved is which of these processes is the one that sleep exists for, the one that if sleep were abolished would fail first and most decisively. The clearance of amyloid beta and the restoration of cognitive function after sleep deprivation suggest that the cleaning process is essential. The lethality of total sleep deprivation, the fact that animals deprived of sleep entirely do not simply become sleepy, but deteriorate and die, suggests that whatever sleep is doing is not optional in any biological sense. But the specific mechanism of that lethality, the precise process whose failure proves fatal when sleep is entirely withheld, has not been definitively identified. There are good ideas, there are partial answers, there are findings that narrow the field and mechanisms that seem on the current evidence more central than others. But the honest account of where sleep science stands is that the broad outlines of what the sleeping brain does have become substantially clearer over the last few decades, while the deeper question, what sleep is fundamentally for, what its loss kills by failing to provide, remains a question without a single agreed upon answer. That this question exists at all, given how much has been measured and discovered, is itself a fact worth holding quietly for a moment. The brain lying in the dark skull is not idle. It runs its slow oscillations through the thalamus and cortex, fires its brief ripples in the hippocampus, moves its cerebrospinal fluid through channels widened by sleeping astrocytes, reorganizes its networks in stage-dependent patterns that researchers are still learning to read. The adenosine that accumulated across the long hours of waking breaks down slowly, steadily, in the basal forebrain, the chemical tide receding with the same patience with which it rose. Outside the laboratory, in any room where someone sleeps, the same biology runs. The EEG trace, if anyone were watching, would show the slow waves arriving in their long intervals, the spindles appearing in brief coordinated bursts, the patterns shifting and repeating across the hours of the night. The cerebrospinal fluid moves through its channels in the dark tissue of the sleeping brain, the glymphatic tide running through the narrow spaces between cells. In the hippocampus, the day's experience replays in compressed form, brief electrical echoes of rooms entered and faces seen, and words learned, firing in the coordinated silence of NREM sleep where the cortex can receive them. This is what the sleeping body is doing, all of it, all at once. The question of why the body is doing it, what purpose was preserved so faithfully across hundreds of millions of years of evolution that no lineage has ever been confirmed to have abandoned it, sits at the edge of what is currently known, a large and genuine open space in the map of biology. Some open spaces in science close quickly. A new instrument arrives, a new measurement becomes possible, and the question resolves into something clear and factual, and no longer open. Other open spaces are different. They persist not because no one is looking, but because the question is genuinely hard, because the answer, when it comes, will require not just a new measurement, but a new way of organizing what is already known. The question of sleep's fundamental purpose has the character of the second kind. The data have been accumulating for decades. The mechanisms are real and well documented, and the question of why a brain as sophisticated as a mammalian brain needs to lose consciousness for a third of each day in order to run biological processes that seem viewed from the outside like they could have been arranged differently, that question remains the kind that a researcher can spend a career working on and still find, at the end, somewhat larger than when they started. The adenosine in the basal forebrain falls through the night. The oscillations move through the cortex in their slow, repeating cycles. The channels in the sleeping tissue stay wide, the fluid moving through them, carrying what the brain has spent and needs to release. And the sleeping brain, electrical and busy and organizing itself in the dark, continues doing what it has always done, what every brain in every sleeping animal has always done, across all the long span of time during which sleep and animals have existed together in the same world. The specific shape of what it is doing becomes clearer with each decade of research. The full reason it has always done so remains, for now, a question as old and as steady and as patient as sleep itself, waiting in the dark for the understanding that has not yet arrived. In a building on the outskirts of Madison, Wisconsin, in the long fluorescent quiet of a research laboratory, a scientist sits with a set of images that do not agree with each other. Each image shows a synapse, the narrow chemical gap across which one neuron reaches toward another. Each image was made with an electron microscope, which sees at a scale far smaller than any lens made of glass, resolving structures no wider than a few billionths of a meter. And what the images show, laid beside each other in sequence, is that the synapse photographed after sleep is measurably smaller than the one photographed before it. The receptor clusters have thinned, the molecular machinery has quietly stepped back. Something happened in the night that the day had not done, and whatever it was, it left a physical mark on the tissue of the brain that a machine could see and measure. This is where the question lives, not in the fact of sleep, which every animal shares, but in the reason for it, the why beneath the stillness, the purpose folded inside the darkness that comes each night. For more than a century, scientists in laboratories on several continents have sat with this question and proposed their answers. The answers are good ones. They are built from real data, from careful experiments, from measurements made in sleeping brains and sleeping bodies across decades of careful work. And none of them alone is enough. The question remains open. It has remained open longer than almost any other question in modern biology, and the openness is not a failure. It is simply the shape the truth takes when it is large enough that no single frame can hold it. Giulio Tononi has spent much of his career inside this question. His laboratory at the University of Wisconsin-Madison has for decades pursued a hypothesis that begins not with what sleep adds to the brain, but with what sleep removes. The logic is careful and specific. During the hours of wakefulness, the brain learns. Every experience, every perception, every thought strengthens the synaptic connections through which neurons communicate. The membrane receptors that detect incoming chemical signals become more numerous. The connections between cells that fire together grow more efficient, more reinforced, more fixed in their patterns. This is how memory forms, and how skills sharpen, and how the accumulated texture of a day of living settles into the tissue of the brain. But there is a cost to this. Stronger synapses require more energy to maintain. A brain whose connections strengthen without limit would eventually exhaust itself, saturating its own circuitry with signal until nothing new could be learned, because everything was already fully potentiated, already written at maximum strength. The brain needs, in some sense, to be reset, not erased. The specific memories must remain, but globally recalibrated, pulled back from the ceiling of its own capacity, so that the next day's experience can register against a background that is not already shouting at full volume. The hypothesis that Tononi developed with his colleague Chiara Cirelli proposes that this recalibration is what non-REM sleep is for. The slow oscillations of deep sleep, the great rolling waves of electrical activity that sweep across the cortex during the early hours of the night, serve, in this view, to globally downscale synaptic strength, not uniformly, not indiscriminately, but in proportion to how strongly each connection was driven during the preceding day, with the result that the relative differences between strong and weak connections are preserved while the overall level drops back to a range the brain can sustain. The landscape of memory is kept, but the volume at which it plays is turned down. In 2017, Tononi and Cirelli and their colleagues published a study that offered direct structural evidence for this idea. Working with rodents, they used an electron microscope to image synapses taken from the cortex of animals after periods of sleep and after equivalent periods of wakefulness. The measurements were painstaking. Thousands of synapses were imaged and measured for size and for the density of AMPA receptors, the primary molecular detectors of glutamate, the brain's main excitatory signal. The results showed that synapses in sleeping animals were on average smaller, and that their receptor densities were lower. The difference was not large. but it was consistent, and it was in the direction the hypothesis predicted. Sleep had physically shrunk the synaptic machinery. The brain had used the night to step back from the edge. The data drew attention. They drew criticism, too, from researchers who pointed out the difficulty of controlling for time of day, body temperature, and the behavioral history of the animals. The debate around synaptic homeostasis is still active, the experimental picture still being refined, but the core observation that the sleeping brain is not maintaining the synaptic status quo, but actively altering it, has held up across replications, and the hypothesis it supports remains one of the most carefully developed accounts of why deep sleep exists. A room away, or 1,000 miles away, or deep inside an earlier decade, a different laboratory is asking a different question. Matthew Wilson's group at MIT wanted to know what the hippocampus does while the animal it belongs to sleeps. The hippocampus is a curved, compact structure in the brain's medial temporal lobe, and it is where the first traces of new memories form, where the spatial layout of a maze just run, or the sequence of sounds just heard, is captured and held, while the slower process of permanent storage elsewhere in the brain has time to complete. During the day, hippocampal neurons called place cells fire in precise spatial patterns as an animal moves through its environment, each cell responding to a specific location, building a neural map of the space the animal is navigating. Wilson and his colleagues recorded from these cells during waking exploration and then continued recording through the subsequent sleep. What they found was that the cells did not go quiet. During NREM sleep, in the hours following a training session in a maze, the same sequences of place cell activity that had fired during navigation fired again, but compressed. A sequence that had taken many seconds to complete during waking was replayed in a fraction of a second during sleep. The same cells in the same order. The pattern of the experience echoing through the resting hippocampus and fast forward. The sleeping brain was rehearsing what the waking brain had done. It was running the memory again, and in doing so, the hypothesis holds, consolidating it, transferring its representation from the temporary storage of the hippocampus to the more durable architecture of the cortex. This is the memory consolidation theory of sleep's function, and its evidence extends well beyond place cells in rodents. Human studies have shown that people who sleep after learning a new motor skill, the sequence of keystrokes for a piano passage, the timing of a finger-tapping task, perform better on that skill the following day than people who stayed awake, and that this benefit is specifically tied to the hours of sleep that contain the most NREM, the most slow-wave activity, the most ripples and spindles. The sleeping brain, in this picture, is not idle. It is working. It is doing, in the dark, the thing that made the skill or the memory permanent. These two ideas, synaptic downscaling and memory replay, are not necessarily in conflict. They may each be describing a different part of the same process. The replay may come first, reactivating the memory traces that are most worth keeping. The downscaling may come after, resetting the overall level while preserving the patterns that replay has reinforced. The brain may be doing both things in the same night, the same sleep, using the same slow oscillations for both purposes, the mechanism serving two needs at once. Or the picture may be more complicated still, and the relationship between these processes may be the next question waiting to be resolved. Meanwhile, in a laboratory at the University of Rochester, another answer was being assembled from a different direction entirely. Macon Niedergard and her colleagues were not looking at synapses or memory traces. They were looking at fluid. Specifically, they were looking at the cerebrospinal fluid that surrounds the brain and spinal cord, the clear, slow-moving liquid that cushions neural tissue against impact and carries away the chemical residue of cellular activity. The brain is metabolically expensive. Its neurons burn through enormous amounts of energy, even during quiet rest, and the byproducts of that energy use accumulate in the narrow spaces between cells. Among those byproducts are proteins, including a protein called amyloid beta, that, if allowed to accumulate in excess, aggregate into the plaques associated with serious cognitive decline. The question Niedergard's group was asking was how the brain cleans itself. The body has a lymphatic system to drain metabolic waste from tissues, a network of vessels running alongside the blood supply, but the brain, walled off behind the tight seal of the blood-brain barrier, has no direct access to that system. For many years, it was not clear how the brain managed its own waste at all. What Niedergard's group found, published in 2013, was that the brain has its own version of a drainage system, one that runs not in separate vessels, but through channels that line the blood vessels already present. Pathways formed by the end feet of astrocyte cells, packed with a water channel protein called aquaporin 4, through which cerebrospinal fluid is driven in slow convective currents. They called it the glymphatic system, a contraction of glial and lymphatic, and the finding that made it relevant to sleep was what happened to this system's activity when the brain's state changed. In sleeping animals, the interstitial space, the gap between cells, expanded by roughly 60% compared to wakefulness. That expansion dramatically increased the flow of cerebrospinal fluid through the glymphatic channels. Fluorescent tracers injected into sleeping animals moved through the brain far more rapidly than the same tracers in waking animals. The cleaning ran fastest while the body was still. Sleep, in this picture, is the maintenance window, the hours in which the brain opens its drainage channels and flushes the molecular residue of the day, including the proteins whose accumulation, over years and decades, may be among the roots of neurodegenerative disease. The clinical implications of this finding moved quickly into wider attention, partly because subsequent human neuroimaging studies showed that a single night without sleep was enough to produce measurable increases in amyloid beta in the brain. The cleaning that should have happened did not happen. The proteins that should have been cleared were still there in the morning, slightly elevated, a small debt on a biological ledger that over a lifetime of poor sleep might compound into something more serious. The glymphatic hypothesis gave sleep a function that felt visceral and immediate, not abstract consolidation of abstract memories, but the physical removal of physical debris, the brain rinsing itself in the dark. And still the question did not close, because none of these hypotheses, however well supported, could fully account for all the data. If sleep existed primarily to clear metabolic waste, one would expect the animals with the highest metabolic rates, the smallest, fastest burning mammals, to sleep the longest, since they would accumulate the most waste in a given interval. The relationship exists, but it is weak and riddled with exceptions. If sleep existed primarily to consolidate memory, one would expect the hippocampus, the organ most central to memory formation, to show the strongest response to sleep deprivation, but deprivation impairs many functions beyond memory, including immune response, hormone regulation, and temperature control, in ways that a memory-only account struggles to explain. The hypotheses are not wrong, they are partial. The immune connection opened through a different set of experiments, in a different institutional setting, with a different set of tools. Stoyan Dimitrov and colleagues examined what happens to T cells, a central class of immune cells, responsible for recognizing and destroying infected or cancerous cells during sleep and during wakefulness. T cells carry on their surface proteins called integrins, which function as molecular adhesives. When an integrin activates, it changes shape, becoming sticky, allowing the T cell to bind tightly to the surface of a target cell and carry out its function. Without integrin activation, the T cell may recognize its target but cannot grip it. The kill cannot happen. The Dimitrov's group found was that integrin activation in T cells is significantly higher during sleep than during wakefulness. The sleeping body is a more effective immune environment. The hours of sleep are not simply passive hours of stillness, but hours during which the immune system shifts into a more aggressive and efficient mode, during which the molecules that allow immune cells to do their work are more fully engaged. Slow-wave sleep, in particular, is associated with elevated production of certain hormones and signaling proteins. Growth hormone rises sharply in the first hours of deep NREM, and the cytokines that coordinate immune responses, including proteins that function as chemical alarm signals between immune cells, are produced in higher quantities during sleep than waking. The sleeping body is in this sense also a healing body, an immune body, a body using the hours of stillness to do the cellular work that the metabolic demands of activity leave less room for. This is not a small thing. The immune system is not a peripheral feature of animal biology. It is the system that determines whether an infection becomes a life-ending event or a temporary inconvenience. An evolutionary connection between sleep and immune function would mean that sleep is not only about the brain, not only about memory and synaptic recalibration and glymphatic clearance, but about the whole organism's capacity to survive microbial challenge. The animals that slept well would have been the animals that fought infection more effectively, that healed from injury more quickly, that lived to reproduce. Sleep, in this view, is woven into survival at a level deeper than any single mechanism can explain. Against all of these ideas, the synaptic, the memorial, the glymphatic, the immunological, there stands a fifth perspective, one that does not seek to identify what sleep does for the brain, so much as to explain when and how long it happens in terms of the world outside. Jerome Siegel, a sleep researcher who has spent years studying sleep in wild living humans and in diverse animal populations, has argued that the standard view of sleep as a biological repair state, whose duration is fixed by internal need, may be missing something important. His ecological niche theory holds that sleep is primarily shaped not by what it accomplishes inside the body, but by what the world outside demands. In this view, sleep is the state an animal enters when the costs of staying active exceed the benefits. At night, for many animals, the risks of moving, of encountering predators in the dark, of burning energy in fruitless foraging, outweigh the returns. The animal that rests is the animal that conserves. Sleep, in this picture, is not a state the brain enters because it desperately needs repair. It is a state the brain enters because remaining active in the dark is for most animals, most of the time, the worse option. The duration and timing of sleep, in this account, are set not by some fixed internal drive, but by the ecological niche the animal occupies, its predation risk, its thermal environment, its foraging schedule, the trade-offs specific to how it lives. Siegel's research in hunter-gatherer communities has shown that sleep in these populations is more responsive to temperature and social cues than the standard laboratory picture would suggest, and that average sleep durations in these groups are somewhat shorter than the 8 hours recommended for Western populations, calling into question the universality of modern sleep prescriptions. His work in wild animals has shown that captive animals sleep very differently from their wild counterparts. that the isolation and inactivity of the laboratory cage may inflate sleep duration in ways that obscure what sleep naturally looks like in an ecologically realistic setting. The natural experiment of the wild may be telling a different story than the controlled experiment of the laboratory. Critics of the ecological niche theory respond with a fact that is difficult to argue past. Total sleep deprivation kills. It kills rats in 2 to 3 weeks. It kills in a way that is not explained by the animal merely being forced to be active in a situation where activity is dangerous. The deprivation itself is the lethal event, not the circumstances that cause it. If sleep were only the state an organism enters when activity is not worth the cost, then forced activity should not kill. It should simply be costly, in the way that many costs are costly. The lethality of deprivation implies that something sleep provides is not optional, not merely energy conserving, not simply ecological positioning. Something essential happens in sleep that cannot be substituted, and when that something is withheld long enough, the body fails. Siegel's account can acknowledge some of this. He does not argue that sleep has no biological function. He argues that the function is served by amounts of sleep more modest than laboratory conditions suggest, and that the duration and structure of sleep are more plastic, more responsive to circumstance than a purely internal drive model implies. The ecological and the biological are not mutually exclusive. The timing of sleep may be set by the world outside, while the necessity of sleep is set by something inside, and the question of how these two levels interact is one the field is still working through. What is left when these 5 perspectives rest beside each other is not a single answer. It is something more like a field of partial answers, each illuminated by real experiments, each touching a different dimension of the same deep structure. The synaptic homeostasis account touches the architecture of learning. The memory consolidation account touches the permanence of experience. The glymphatic account touches the chemistry of neural maintenance. The immunological account touches the body's defense against infection and injury. The ecological account touches the relationship between the animal and the world it navigates. None of them is false. None of them is complete. There is a specific quality to this scientific situation that is worth staying with for a moment. Biology has solved many of its old mysteries. The mechanism of inheritance was open for most of human history and was resolved in the middle of the 20th century. The process by which cells generate usable energy from food was similarly resolved, the pathway mapped, the enzymes named. These were not simple problems, and solving them required generations of work. But they were solved. The question of why sleep exists, why every animal studied must enter a state of elevated vulnerability and diminished awareness for a significant portion of every day, has not been solved in this way. The data keep accumulating, the hypotheses keep being refined, and the mystery keeps revealing new dimensions as it is approached. Part of what makes this situation unusual is the nature of the evidence. Sleep deprivation experiments show that sleep is necessary, but they cannot directly show what specifically makes it necessary. If a factory is shut down for a week and everything that depended on it fails, the shutdown proves the factory was essential, but does not specify which of the factory's many processes was the critical one. Sleep may be necessary for memory, and necessary for waste clearance, and necessary for immune function, and all of these needs may have co-evolved into a single unified state, because an animal cannot afford to take separate recovery periods for each of them separately. The unification of multiple functions into a single behavioral state may itself be part of the answer. Sleep as the convergence point of several different biological necessities that evolution found it efficient to satisfy simultaneously. This kind of convergence would explain something that is otherwise puzzling about the comparative biology of sleep. Animals with very different brain structures, very different metabolic rates, very different ecological histories, all share the same basic behavioral signature of sleep: the quiescence, the elevated threshold, the rebound when sleep is prevented. If sleep existed for a single specific function, one might expect that function to vary more across such diverse lineages that some animals would have evolved alternative ways of achieving the same end. The near universality of sleep, its resistance to evolutionary elimination, its persistence across 500 million years of independent lineages, suggests that whatever it accomplishes is not easily replaced. The functions may be multiple, and they may be so deeply interlocked that removing sleep means removing all of them at once. In a quiet corner of the immunological literature, a finding sits that illustrates this interlocking character. The cytokines that promote sleep, the immune signaling molecules that rise during infection and drive the extended sleep of a sick body, are also molecules that promote the slow-wave activity of NREM sleep in a healthy brain. Sleep and immune function share a molecular vocabulary. The signals that the immune system uses to call for more sleep are the same signals that deepen the sleep that is already happening. The system that defends the body and the system that regulates rest are not separate systems communicating across a gap. They are entangled systems using shared language, and the entanglement is deep enough that it looks like coevolution, like two biological processes that have been optimizing together for a very long time. The same entanglement appears at the interface of sleep and memory. The slow oscillations of NREM sleep, the large coordinated waves that sweep the cortex in the deep hours of the night, trigger the precise sequence of electrical events in the hippocampus and thalamus that is associated with memory replay and memory transfer. The wave arrives, the hippocampus fires in its compressed replay. The thalamus adds its spindle, and the dialogue between structures carries the day's experience one step further toward permanence. This is not a process that happens alongside sleep. It is a process that sleep enables, that the specific electrical state of NREM makes possible in a way that wakefulness does not. The function and the state are inseparable. Tononi and Sorelli's synaptic downscaling happens in the same electrical landscape. The slow oscillation is the mechanism. During the down state, the moment of near silence that alternates with the burst of activity in each slow wave, the synaptic machinery is given a brief window of reduced excitation, and during those windows, the molecular processes of receptor removal and synaptic retraction can proceed. Without the oscillation, the window does not open. Without sleep, the oscillation does not happen. The function requires the state, and the state somehow serves multiple functions at once, using the same electrical infrastructure for different biological purposes in overlapping time. A researcher in this field, sitting with the accumulating evidence, might describe the situation as standing before a room with 5 doors, each of which opens onto a real and well-lit corridor, and each corridor leads in a slightly different direction, and none of them has yet been followed to the end. The corridors may converge in some deeper chamber that has not yet been reached. They may not. The evidence does not currently specify which of these is true, and the researchers who have spent careers exploring one corridor or another have not yet found the place where the corridors meet. There is patience in this work. Sleep science is not a young field. The basic electrical signatures of sleep were described in the first half of the 20th century, and researchers have been building on those foundations for decades. The patience is built into the practice, Sleep research requires sleeping subjects, and sleeping subjects require time, and the slow rhythms of the biological night impose a tempo on the work that cannot be hurried without distorting the phenomenon being studied. You cannot rush a slow oscillation. You cannot sample memory consolidation in a few minutes. The science moves at the pace of sleep itself, which is to say, it moves slowly and builds over years, and does not arrive at its conclusions in a single bright moment. This is perhaps part of why the question remains open. The tools needed to see what is happening in the sleeping brain at the cellular and molecular level have only recently become available. The ability to record from thousands of neurons simultaneously, to image cerebrospinal fluid flow in living tissue, to measure receptor density at individual synapses. These capabilities arrived in the last decade or two, and the data they are generating are still being integrated into the theoretical frameworks that predate them. The question has been asked for a long time. The means of answering it precisely are new, and the answers they are producing are still arriving. What the field does agree on is simpler and more fundamental than any of the specific hypotheses. Sleep matters. The evidence for this is not one study or one experiment, but a convergence of evidence from every level of analysis, from the lethality of total deprivation to the molecular conservation of sleep regulatory genes across 600 million years of evolution, to the behavioral evidence that every animal studied enters something recognizable as sleep and pays a real cost to do so. Something in sleep is essential. What that something is, whether it is one thing or many things, whether the mechanisms in the mouse are the same as the mechanisms in the sparrow, whether the function identified in the hippocampus is the whole story or a fraction of a larger picture, remains genuinely open. 5 frameworks rest beside each other in the literature, each with its evidence, each with its gaps, each representing years of careful work by researchers who have devoted their professional lives to the inside of this question. The synaptic account, with its electron microscope images of smaller, recalibrated receptor clusters. The memory account, with its compressed place cell replays firing in the sleeping hippocampus. The glymphatic account, with its slow fluid currents carrying molecular residue through the expanding interstitial space. The immunological account, with its sleeping T cells and their activated integrins gripping targets more effectively in the night. The ecological account, with its wild animals sleeping on schedules shaped as much by darkness and danger as by internal biological need. They do not resolve into a single picture. They sit together in a kind of productive tension, each illuminating what the others do not, each serving as a corrective to the other's overreachings. The field is not stalled. It is doing what science does when it is working well, accumulating constraints on a problem whose solution is not yet visible, narrowing the space of possible answers, building toward a clarity that may arrive in 5 years or 50, in the work of someone who has not yet begun. And the question itself, why sleep? Why this? Why every animal? Why no lineage has escaped it? Rests where it has rested for a long time. Open, attended, surrounded by good evidence and careful thought, and the slow accumulation of measurement after measurement in the dark hours of the laboratory night. 5 corridors, each well lit, 5 partial answers, each real, and somewhere in the structure that connects them, the full shape of the answer, waiting in the way that deep things wait, without impatience, without urgency, in the same unhurried dark where every animal, in every lineage, on every continent, lies down each night and closes its eyes. In the dark water off the coast where the shelf drops away, and the surface light dissolves into blue and then into nothing, a dolphin moves in long, slow arcs through the middle depth. One eye is open, the other is closed, and in the brain behind those two eyes, two entirely separate things are happening at once. One hemisphere lit with the low, steady activity of wakefulness, tracking the pressure changes in the water, holding the body's course, timing the slow rise toward the surface and the breath that comes with it, and the other hemisphere sunk into the long electrical quiet of sleep, its neurons cycling through rhythms that no waking state can reach. The animal is whole. The animal is moving. The animal is, in the most exact biological sense, simultaneously asleep and awake. This is not a metaphor. It is a measured, documented, electrical fact, first recorded by a Russian researcher named Lev Mukhametov in the 1970s, who placed electrodes against the skulls of dolphins swimming freely in their tanks and watched the traces come back showing, unmistakably, two hemispheres on different schedules, one dark while the other ran. What he was looking at had no precedent in the sleep literature. It did not fit the framework built from studying mammals that sleep with both hemispheres together, that lay down and close both eyes and pass through the same stages at the same time in the same brain. A dolphin could not do that. A dolphin that fell into the bilateral unconsciousness of deep mammalian sleep would stop breathing and sink. So the brain had found another way. It had divided itself. It had made sleep something that one half could accomplish while the other half continued to live in the world. And then it had reversed the arrangement, and then reversed it again, and across a night the animal had rested and navigated and surfaced and breathed without ever once surrendering to the full stillness that most mammals require. The sleeping eye faces downward toward the dark below. The waking eye faces outward, watching. What this tells researchers who study sleep across species is that the brain's requirement for whatever sleep provides is precise enough to be managed hemisphere by hemisphere, timed and allocated and tracked. The debt accumulated in one structure paid off while the other keeps working. Sleep, in other words, is not simply unconsciousness. It is not just the body switching off. It is something specific enough that the brain can portion it out, something necessary enough that even an animal shaped by millions of years of life in open water where stillness means death cannot be without it, and something flexible enough that biology found a way to deliver it anyway, in a form that the ocean would allow. Migratory birds carry the same capacity. The white-crowned sparrow, small and brown and unremarkable to look at, crosses thousands of miles of open land twice each year, pressing through the night sky in long, unbroken flights that would seem to make sleep impossible. In the weeks surrounding migration, its total sleep time drops by more than half, not through deprivation, but through reorganization, the brain shifting toward briefer, more frequent unihemispheric episodes, snatching rest in fragments so small they leave barely a visible trace in behavior. Research by Niels Rattenborg at the Max Planck Institute has shown that these birds suffer almost none of the cognitive impairment that mammals display under equivalent sleep restriction, as though the sparrow's brain has learned to spend its sleep more efficiently, to extract from a shorter session what a mammal would need a longer one to accomplish. The debt is real, the sparrow pays it, but it has found a currency the night sky will accept. These are not the strange exceptions at the margin of what sleep is. They are among the most revealing things known about it, because they show that whatever sleep does, the brain guards it, protects it, restructures the entire architecture of a life to keep delivering it, even when the world makes it dangerous or difficult or nearly impossible to obtain. The dolphin does not decide to sleep with one hemisphere. The sparrow does not choose its migration schedule around its cognitive performance. The requirement came first, deep in the biology, and the behavior built itself around that requirement across vast spans of evolutionary time, finding every available workaround, every possible accommodation, every form that rest could take, and still accomplish what rest needs to accomplish. And across the animal kingdom, those forms are stranger and more varied than any single framework built from watching mammals sleep in laboratories could have predicted. A fruit fly rests on the wall of its enclosure, motionless, its legs folded, its small body pressed to the surface. The lights in the laboratory are dim. The temperature has dropped slightly, as it does each evening in the rhythmic cycle the researchers maintain to approximate a natural day. And the fly, with its roughly 100,000 neurons and its lifespan measured in weeks rather than years, has entered a state that meets every behavioral criterion used to define sleep in far larger, far more complex animals. It is quiescent. Its arousal threshold has risen measurably. A stimulus that would send it moving during wakefulness produces no response now, or a slower one. If kept awake artificially through the night, it will sleep more deeply and more durably the following day, exactly as a sleep-deprived mammal does. The homeostatic rebound as precise and proportional in this creature of a few milligrams as it is in a dog or a primate. The fruit fly does not merely resemble a sleeping animal. It possesses specific neural circuits that regulate sleep, identifiable populations of neurons in structures called the mushroom bodies, and the fan-shaped body that drive the transition into rest and govern its depth and duration. When researchers manipulate these circuits, the fly's sleep changes in predictable, measurable ways. When the circuits promoting sleep are activated artificially, the fly sleeps more. When they are suppressed, it sleeps less and accumulates a deficit that demands repayment. The system has the same logic as the mammalian sleep system, running in a brain small enough to sit on the head of a pin, in an animal that will live and die in less time than a single human sleep study takes to complete. The worm is smaller still, Chinorhabditis elegans, transparent and a millimeter long, lives in soil and laboratory dishes, and has been the subject of more genetic research than almost any other animal alive. Its entire nervous system consists of 302 neurons, every single one identified, named, and mapped. And yet this animal, with its handful of cells and its ancient lineage, enters a sleep-like state called lethargus at each transition between larval stages, a state governed in part by a neuropeptide called APTF1, whose counterpart in mammalian brains performs similar sleep regulatory functions. The molecule is old, older than the brain, older than the synapse as mammals know it. The molecular instruction to rest reaches back to before there were creatures with heads to sleep on. What this depth of conservation means is that sleep is not something that mammals invented and other animals approximate. It is something that the first animals needed, for reasons that were already written into the chemistry of early nervous tissue, and that every lineage since has carried forward and elaborated and specialized according to its own circumstances, but never once discarded. The dolphin's unihemispheric solution and the sparrow's migratory compromise, and the fruit fly's mushroom body circuits and the worm's lethargus are not different things loosely grouped under one label. They are variations on a theme so old and so necessary that it has never in any confirmed lineage been abandoned. A sleeping bat hangs inverted in the dark of a cave, its wings folded against its sides, its body temperature dropping toward the cold of the surrounding stone. The little brown bat will spend close to 20 hours of every 24 in this state, more time asleep than any other mammal commonly studied, its metabolic rate falling so low during the deep phases that its small heart beats only a handful of times a minute. An elephant, by contrast, sleeps approximately 2 hours in every 24, resting briefly in the small hours of the night, sometimes standing, sometimes lying down in the leaderite dust of the savanna, in its vast body requiring only the shortest interval of rest before rising again to spend the remaining 22 hours feeding and walking and navigating the social complexity of its herd. Between these two animals lies a tenfold range of sleep need, and within that range sits every other mammal studied, the horse sleeping 3 hours, the lion sleeping 12, the human sleeping 8, the ferret sleeping 14, and the question of what determines where any given species falls within this range has resisted every analysis brought to bear on it. Body mass explains some of the variation, but not much. Metabolic rate explains some, but not enough. Predation risk shapes sleep architecture. Prey animals tend toward lighter, more fragmented sleep, their arousal thresholds kept closer to wakefulness, but it does not simply determine total sleep time in a linear way. Brain size, social structure, diet, habitat, thermoregulatory strategy, none of these variables, alone or in combination, accounts for more than a fraction of the variance that the data contains. The bat sleeps 20 hours because the bat needs 20 hours. The elephant sleeps 2 because 2 is what the elephant requires. But what exactly those hours are providing, and why the requirement differs so enormously between two warm-blooded mammals with chambered hearts and fur and nursing young, this remains, for now, without a clean answer. The data is real. The difference is real. The explanation has not yet arrived in a form that satisfies the researchers who spend their lives looking for it. Sleep duration is not merely a trait like coat color or the shape of a tooth. It is a variable that speaks directly to the question of what sleep is for, because if sleep serves a single universal purpose, it is difficult to explain why an elephant needs so little of it, and a bat needs so much. The difference implies either that the purpose scales with something not yet identified, or that sleep serves several purposes simultaneously, and different species lean on different ones, or that the ecological niche theory is partly right, and the bat's long sleep reflects not a biological need, but a behavioral solution to a world where daytime activity carries more cost than benefit for an animal that hunts by echolocation in the dark. Jerome Siegel, whose ecological niche theory argues precisely this, points to data showing that some of the longest sleepers are animals with little predation risk and limited waking opportunities, large carnivores and insectivores that eat quickly and have little else to do. The sleep on this view is partly strategic in activity, the body conserving energy and avoiding risk during the hours when activity would offer diminishing returns. Critics of this view point to the lethality of total sleep deprivation and ask why any state that is primarily strategic in activity should be lethal to withhold. A fasting animal does not die of not eating in 2 weeks if food is freely available. A restrained animal does not die of being kept awake through the intervention of something other than the withholding of food. But a sleep-deprived rat in a laboratory, with all the food and water it can consume, will still deteriorate and die, its temperature regulation failing, its immune barriers breaking down, its weight dropping despite the calories it is consuming, as though something the body cannot get from eating or drinking or resting quietly is being withheld, something that only the sleeping state itself delivers, and that the body cannot survive without for long. These two views, sleep as primarily ecological versus sleep as biologically essential, in ways irreducible to rest, have not been resolved. They have simply continued to sit beside each other in the literature, each with real evidence, each with real limits, neither yet capable of absorbing the other. Far out on the evolutionary tree, past the vertebrates and the arthropods and the mollusks, past the cnidarians whose jellyfish trail their tentacles through warm surface water, lies the upside-down jellyfish called Cassiopeia. It rests on the seafloor in shallow tropical lagoons, its bell facing downward, its fringed oral arms reaching upward through the water like pale flower petals, pulsing slowly to circulate the water over the symbiotic algae living in its tissue. Cassiopeia has no brain. It has no centralized structure of any kind that would qualify as a nervous system in the sense mammals have one. It has a diffuse net of neurons spread across its body, capable of coordinating the rhythmic contraction of its bell, but not of doing much else that researchers can currently measure. And yet in 2017, a team at Caltech documented that Cassiopeia sleeps. At night, its pulsation rate drops, not slightly, measurably, reliably, consistently, the same pattern each night, the same recovery each morning. A stimulus that prompts a strong response during the day produces a weaker, slower response during the night hours. The arousal threshold elevated in the dark, exactly as it is in a sleeping mammal, a sleeping fly, a sleeping worm. When the researchers kept Cassiopeia awake artificially through the night, applying perturbations that disrupted its rest, the jellyfish the following day showed increased quiescence, a homeostatic rebound, the same debt and repayment dynamic that defines sleep as sleep rather than simply inactivity, present in an animal with no brain and no centralized processing of any kind, in a body that diverged from the lineage leading to vertebrates more than 500 million years ago. 500 million years. The number lands slowly when held quietly in the mind. Whatever sleep is doing, it was doing it before there were fish, before there were limbs, before there were eyes, in the form that vertebrates carry them. It was doing it when the ancestor of Cassiopeia and the ancestor of every mammal alive were still the same animal, or something close to it, drifting in a warm, shallow sea, simple and small, and already, somehow, in need of rest. The discovery of sleep in Cassiopeia did not solve the mystery of why sleep exists. It deepened it. Because if sleep requires no brain to manifest, if the behavioral logic of quiescence and elevated threshold and homeostatic rebound can run in a diffuse nerve net spread across the body of an animal without a head, then whatever sleep is serving is not solely a function of the centralized brain. It is not just memory consolidation, which requires a hippocampus. It is not solely glymphatic clearance, which operates through a cerebrospinal system that Cassiopeia does not possess. Something older than brains needs this rest. Something at the level of the nerve itself, or the cell, or the tissue, or the gap between firing and recovery. Something that was already present before the nervous system gathered itself into a center and began to process and remember and dream. The implications of this reach in every direction at once, and researchers who think carefully about them tend to arrive at the same quiet conclusion. There may not be a single answer. There may be a set of answers, each real, each operating simultaneously, some older than others, some shared across all animals, and some elaborated in specific lineages. And the question of why sleep exists may be like the question of why the heart exists. Not one answer, but several, beating together, inseparable from each other in the living thing that relies on all of them at once. A migratory bird rests for a moment on a branch at the edge of a continent. The sky ahead is open water. The distance to the far shore is 3 days of unbroken flight. In its brain, the neural circuitry governing the timing of sleep and the depth of rest is already reorganizing itself for what is coming, shortening the cycles, distributing the rest across more frequent but briefer episodes, preparing the bird for a crossing that should be, by any measure derived from studying mammals, biologically impossible. It is not impossible. The bird has made this crossing before. Its parents made it. The lineage has made it for longer than the mountain ranges it flies over have existed in their current form, and the sleep system that makes it possible has been refined across all of that time into a form that no laboratory studying only nocturnal mammals in quiet cages would have predicted. The sparrow does not know this. It does not know the distance. It does not know the biology. It sits for a moment in the last of the evening light, and then it rises, and the dark takes it, and somewhere inside its small skull, the two hemispheres begin their ancient alternation, one resting, one watching, one resting, one watching, and the miles pass beneath it in the dark. There is something quietly extraordinary about what comparative sleep research has revealed across the last half-century, not because it has answered the central question, but because of the shape of what it has found. Every species studied sleeps, or enters a state that sleep's own defining criteria mark as sleep. Every lineage that has been followed deeply enough has shown that this state is regulated, tracked, and recovered when lost. No confirmed case of a naturally sleepless animal has emerged from the hundreds of species examined. The dolphin, the sparrow, the fly, the worm, the jellyfish, none of them can do without it. All of them have made whatever accommodation their lives demanded to keep receiving it. And the deeper the research goes into smaller nervous systems, into older lineages, into animals that share fewer and fewer of the structures through which researchers first understood sleep, the clearer it becomes that the need runs below everything, to a depth that current understanding has not yet reached. A harbor seal floats at the surface with its nostrils barely above the waterline, sleeping in slow vertical spirals, its body rotating through a long arc that brings it back to the air just before it would need to breathe. In the deep ocean, sperm whales have been photographed resting in pods near the surface, drifting in loose clusters, motionless and vertical, their vast bodies hanging in the blue like irregular columns of dark stone, breathing at long intervals, returning each time to stillness. These are animals shaped by the same evolutionary pressure that shaped the dolphin's split hemisphere solution, and they have arrived at slightly different forms of the same accommodation, ways of getting the rest that cannot be foregone while living in a medium that makes rest dangerous. The ocean does not become less dangerous because the whale is tired. The sky does not become less vast because the sparrow needs to close one eye. Predators do not pause because a rabbit has entered its 50th brief shallow bout of the night, its posture softened, but its ears still angled outward, its arousal threshold raised just far enough that small sounds pass unremarked, but large ones still arrive with enough force to send it moving. The world does not accommodate sleep, and sleep accommodates the world, finding whatever form the world will permit, pressing itself into the available shape, insisting on being received by bodies that cannot function indefinitely without it, regardless of what the circumstances of those bodies require them to risk. And that insistence, quiet, biological, non-negotiable, is the thing that strikes sleep researchers most when they step back from the details of any single experiment or any single species and look at the whole of what has been accumulated. Sleep is not optional in the sense that any confirmed lineage has ever made it optional. It is not reducible to mere inactivity, because inactivity does not produce the rebound that only lost sleep produces. It is not solely a mammalian phenomenon, or a vertebrate one, or even a nervous system one, in the sense that requires a brain. It appears to be something the body needs at a level so fundamental that it has been conserved across every major divergence in the history of animal life. Carried from the earliest nervous tissue forward through every transformation that tissue has undergone, from diffuse net to ganglion to centralized brain to the thalamocortical architecture, through which a sleeping human cycles through its stages each night. And after all of that time, after all of that conservation, after all of the research and the recordings and the deprivation experiments and the genetic analyses and the electron microscopy and the imaging studies, the question of exactly what sleep does, that justifies all of this, the vulnerability, the lost foraging time, the metabolic cost of maintaining the machinery that sleep requires, remains genuinely and cleanly open, not almost answered, not nearly resolved, open in the way that the deepest scientific questions are open, not for lack of trying, but because the thing being asked is harder than it looks, because every answer that seems sufficient contains within it a smaller, harder question that the next experiment reveals, because the mystery keeps deepening rather than narrowing as the tools improve and the data accumulates. A jellyfish rests on the seafloor in the dark of a tropical lagoon, its bell pressed down into the soft sediment, its oral arms still. The water above it is black. The pulsation has slowed. If a perturbation arrived now, a shadow, a current, a touch, the response would come, but it would come more slowly than it would in the afternoon, more slowly than it would in the morning, because the animal is in the borrowed stillness of its nightly rest. The ancient rhythm, running as it has run in this lineage for longer than the continents have held their current shapes. No brain governs this. No hippocampus is replaying the day. No thalamic spindles are rising and falling through a cortex. There is only the nerve net and the rhythm it keeps, and the debt that accumulates when the rhythm is disrupted, and the recovery that follows when it is allowed to run. Whatever sleep is in its deepest form, its most reduced and ancient form, it is here. It was here before it was anything else. And what it is, what it does, what it repairs, what it accomplishes in the slow dark hours that cannot be accomplished any other way, remains the question that every sleeping animal, alive, from the whale to the worm to the quietly resting human, poses and answers nightly without ever explaining. The lagoon holds the jellyfish. The dark holds the lagoon, and the night continues, slow and wide and full of things at rest. In a single cell, inside a liver, in the body of an animal lying still in the dark, a molecular clock is turning. Not turning the way a watch turns, with teeth and springs and the tick of a metal hand, turning the way seasons turn, through accumulation, through threshold, through the slow inevitability of a chemical tide that rises and then falls back on itself. Two proteins bind together in the cell's nucleus and reach out toward a strand of DNA, and what follows from that touch is a sequence of events so finely calibrated that it repeats itself within minutes of the same interval, day after day, for the whole life of the animal. The proteins are called clock and BMEL1, and the loop they initiate, one protein complex opening a gene, the gene-producing proteins that eventually close the complex down, the complex fading, the gene opening again, completes itself in roughly 24 hours. Not roughly in the way an estimate is rough, roughly in the way a tide is rough, close enough that the body can use it as a clock, and precise enough that drift across a week amounts to less than an hour. This loop runs in the liver. It runs in the heart. It runs in the lungs and the skin in the fat, in the bone, in cells that have never fired a single nerve signal and never will. The brain has its own master clock, a small paired structure sitting just above the point where the optic nerves cross, taking its daily reset from light falling on the retina. But below that master, across every tissue, the same molecular oscillation is running independently, slower or faster in different organs, held in coordination by signals from the center, but capable of running alone in a dish of cells kept warm in a laboratory. Cut a liver away from the body and keep it breathing, and its cells will keep time for days. The body is not one clock, it is hundreds of millions of clocks, each ticking on the same inherited rhythm, each one a copy of a mechanism so old that its essential architecture is recognizable in organisms alive today that diverged from the human lineage more than 1 billion years ago. Single-celled algae cycle through predictable patterns of activity and rest. Fungi oscillate. Cyanobacteria, some of the oldest living things, keep a circadian rhythm so robust it can be reconstituted in a test tube from just 3 purified proteins. What this means is that before there were animals, before there were nervous systems, before anything slept in any way a researcher would recognize, something in living matter was already counting the hours. The circadian clock does not cause sleep, but it sets the window. It negotiates constantly and chemically with a second system, the pressure that builds in the brain, simply from being awake. The longer an animal goes without sleep, the greater the accumulation of a molecule called adenosine in the fluid surrounding the brain's waking centers. Adenosine is a byproduct of the work neurons do, a small, unremarkable chemical left over when cells burn their fuel, and its concentration in the basal forebrain rises through every waking hour like water rising in a sealed room. As it rises, it binds to receptors on the neurons that keep the brain alert, and one by one, those neurons grow quieter. It binds to receptors on neurons that promote sleep, and those neurons, released from inhibition, grow more active. The pressure is not a signal from any one part of the brain. It is a property of the brain as a whole, a distributed vote, cast in chemistry, accumulating across every hour the animal remains awake. These two systems, the circadian clock and the adenosine tide, were described together in a model proposed by a Swiss researcher named Alexander Borbély in the early 1980s. He called them Process C and Process S, the circadian drive and the homeostatic pressure, and described sleep as the state the brain enters when the pressure of S rises high enough to override the timing signal of C. The model was elegant. It was also, in the way that good models often are, a description of mechanism without an answer to the deeper question underneath it. It explains when sleep comes. It does not explain what sleep is for. Somewhere inside a sleeping mouse, in a small tuck of tissue, in the hippocampus, a neuron fires. It fires in a pattern. The pattern is not random. It is a sequence that played out hours earlier, when the mouse was moving through an environment it had never encountered. A new maze, a new set of turns, a new arrangement of spatial landmarks. The neuron that fires now is a place cell, the kind of cell that activates when an animal occupies a particular location in physical space, and the sequence it is now producing in sleep is a compressed replay of the sequence it produced when the mouse was actually moving. The replay is faster, perhaps 10 or 20 times the speed of the original, and it does not happen alone. At the same moment that the hippocampal replay is running, bursts of activity are arriving from the thalamus, brief, rhythmic oscillations called sleep spindles, 8 to 15 cycles per second, each one lasting less than 2 seconds. And beneath both of these, the cortex is generating its own slow rhythm, a long wave of activity followed by a long wave of near silence, cycling once every second or two, the breath of the sleeping brain. These three rhythms, the hippocampal replay, the thalamic spindles, the cortical slow oscillation, are not independent. They are coupled. The replay tends to happen inside the slow oscillation's active phase. The spindles tend to arrive at the moments between replays. Something is coordinating them, and what is being coordinated, the best current picture suggests, is the transfer of information from a temporary store to a more permanent one. The hippocampus holds what happened today. The cortex holds what has been integrated across many days, many experiences, the slow accumulation of a life. Sleep may be the time when the hippocampus reads its notes aloud and the cortex listens. This is not certain. The hypothesis has real evidence, and it also has limits. The replay is real. It has been measured many times in rats and in humans, in visual cortex and motor cortex and olfactory regions. The coupling between the three rhythms is real. What remains open is the question of whether this replay is necessary for memory consolidation, or whether it is one mechanism among several, or whether the same oscillatory patterns serve functions beyond memory that have not yet been identified. The sleeping brain is doing many things at once, and the tools available to watch it are still young. In a channel in the wall of a neuron, a gate is opening and closing with the rhythm of the night. The channel is made of proteins folded into a tube no wider than a few atoms across, spanning the membrane that holds the neuron's interior apart from the fluid outside it. When the gate opens, calcium ions flow inward, a small current, a brief change in the electrical balance across the membrane. When the gate closes, the neuron resets. In the particular cells of the thalamus that drive slow-wave sleep, these gates open and close in a rhythm that the cell cannot escape. A burst of firing, a hyperpolarization that silences the cell, and then a rebound burst triggered by the same channels that cause the silence. The cell oscillates not because anything is commanding it to oscillate, but because of the physics of its own membrane. The channel type responsible is called a T-type calcium channel, and mice in which the gene encoding it has been deleted do not produce normal slow-wave oscillations. Their sleep is lighter. Their memory consolidation, measured on standard behavioral tests, is impaired. This is a different kind of answer to the question of what sleep does. Not a behavioral answer, not an ecological answer, but a biophysical one. Sleep is what happens when a particular class of ion channel, in a particular population of cells, drives a particular oscillatory pattern that the brain requires. Remove the oscillation, and the animal is diminished in specific and measurable ways. The channel is not optional. The rhythm it produces is not optional. Whatever sleep is for, this mechanism is part of how it is delivered. The gene encoding the T-type calcium channel belongs to a family of genes found across many species, from worms to mammals, each producing channels that shape the electrical behavior of nerve cells in ways that are tuned to the particular nervous system they inhabit. The genetic logic of sleep, looked at closely, is not a human invention or even a vertebrate one. It is much older than that. A fruit fly in a laboratory rests in the dark, its small body motionless on the wall of a vial. It will rest this way for several hours. If something disturbs it, a tap on the glass, a brief vibration, it takes longer to rouse than it would in the middle of the day. If it has been kept awake artificially, it will rest for longer than usual when the interference stops, as if paying back a debt. These are not metaphors. They are measurements, the same behavioral criteria that define sleep in mammals, applied to an animal with a brain smaller than a pinhead and satisfied fully. The fly sleeps. In flies with a mutation in a gene called shaker, the sleep is shorter. The shaker gene encodes a potassium channel, a different kind of membrane gate, and when it is disrupted, the neurons that should enter rest do not do so efficiently. The mutant flies live shorter lives. They perform poorly on learning tasks. The mutation's effects reach beyond sleep into cognition and health in ways that closely parallel the effects of sleep deprivation in larger animals. And the potassium channel that the shaker gene produces in the fly has a structural relative in the mammalian brain, a channel called KV1. 2. That plays analogous roles in neuronal excitability and is found in the same kinds of inhibitory circuits. Across 600 million years of separate evolution, the molecular machinery of sleep has not been discarded. It has been maintained, modified, built upon, elaborated into new forms, but never abandoned. What this says about the function of sleep is indirect but powerful. Natural selection is a process of elimination over geological time. Whatever does not help an organism survive and reproduce tends slowly to be lost. Whatever persists across vast stretches of time, through countless environments and countless threats, tends to persist because its loss would cost more than its continuation. The molecular sleep machinery in the fly and the molecular sleep machinery in the mammal did not stay similar by accident. They stayed similar because something in what they do is necessary, and necessary in the same deep way, in bodies that share almost nothing else of their design. But the shaker fly offers something more specific than this general argument. It connects sleep to neural excitability, to the behavior of individual cells, to the question of what a neuron that is not regularly inhibited will eventually do. A neuron that fires without rest degrades. Its molecular machinery for repair runs behind the pace of its use. The proteins that make up its synapses accumulate damage. The connections it makes with other neurons drift out of calibration. Some researchers have proposed that this cellular wear and the repair that sleep enables is itself the answer to the question of sleep's function, that sleep exists not to serve the brain's higher purposes, but to maintain the health of its most basic units. Others have proposed that these are the same thing, that maintaining the health of individual neurons is precisely what makes memory and cognition and the higher purposes possible. Deep in the brain's interior, in the narrow channels that run alongside blood vessels in the sleeping cortex, fluid is moving. The channels are formed by the end feet of support cells called astrocytes, which wrap around blood vessels and press their membranes into a close embrace, leaving a thin space through which cerebrospinal fluid can travel. During wakefulness, this space is narrow. During sleep, something changes. The astrocytes contract slightly, or the space between cells expands for reasons still not fully understood, and the gap widens by roughly 60%. Through this wider channel, cerebrospinal fluid flows more freely, driven by the pulse of the arteries and the slow oscillation of the sleeping brain itself, and as it flows, it carries away the molecular residue of a day's neural activity. Broken protein fragments, oxidized lipids, the accumulated byproducts of billions of cells having burned their fuel for the past 16 hours. Among the molecules this fluid carries is amyloid beta. In a healthy brain, the overnight clearing keeps pace with the overnight accumulation. In a brain that does not sleep enough, the balance shifts. Amyloid beta concentrations in the fluid bathing the brain rise measurably after a single night without sleep. The brain has mechanisms for clearing this molecule during waking hours, but they are slower and less thorough than the mechanisms available during deep sleep. The particular name for this clearance system is the glymphatic system, after the glial cells that form its channels, and the aquaporin 4 proteins embedded in the astrocyte membranes that help drive the fluid flow. Whether clearing amyloid beta is the primary function of sleep, or a secondary benefit, or one of many simultaneous processes that sleep enables, is not settled. The clearance is real. Its role in the full story of why sleep exists is part of what remains open. What is not open is that the brain during sleep is not idle. The EEG trace of a sleeping human, running beside the trace of a wakeful one, is not a flat line beside a busy one. It is a different kind of business, slower in some phases, startlingly active in others, organized in ways that still carry more information than the tools currently available can fully read. In a research building somewhere, the genetic material of a small family has been sequenced. 2 members of this family function fully on roughly 6 hours of sleep per night, not 6 hours of adequate sleep, 6 hours of what appears to be genuinely sufficient sleep, without the performance degradation seen in most people who restrict themselves to the same duration. They wake without alarms. They are alert and cognitively capable through long days. The sleep they get, measured in a laboratory, looks like normal sleep on every scale available, but they need less of it. When researchers mapped their genomes, what they found in both family members was a single difference in a gene called DK2. One amino acid changed from the standard human sequence, a substitution so small it would be invisible to any technology that read the gene by eye. DEC2 is a transcription factor, a protein that regulates the activity of other genes, including some involved in the timing and duration of sleep. The single substitution appears to make the protein slightly less effective at suppressing the wake-promoting systems of the brain, and the result, across a whole human life, is a genuine and stable reduction in sleep need. This finding was important, not for what it said about this family, but for what it said about the genetic architecture of sleep need in general. Sleep duration, how long any individual animal seems to need, is not fixed at the species level in a simple way. It varies from person to person, and some of that variation appears to be inherited. Subsequent research identified other variants in other genes, including ADRB1 and NPSR1, each associated with shorter habitual sleep in the people who carry them. The genetic landscape of sleep need, looked at from a distance, appears to be complex, many genes each contributing a small adjustment, adding or subtracting minutes from the total, shaped over evolutionary time by pressures that are not yet fully understood. The vast majority of people who describe themselves as naturally short sleepers are not. Sleep restriction studies, in which people are kept to 6 hours or fewer per night in controlled laboratory conditions, show consistent and measurable declines in cognitive performance over days, declines that the sleepers themselves often do not perceive, because the awareness of impairment also declines with sleep loss. The genuine short sleepers, those who carry identifiable genetic variants and show no measurable impairment on objective testing, are rare. They are worth studying precisely because they mark the outer edge of what sleep can be, and because the genes that permit them to function on less may point toward the mechanisms that determine how much sleep is enough for any individual. But even knowing the variants, even identifying the genes, does not explain what sleep is doing in the hours it occupies. A map of the genetic controls on sleep duration is not the same as an explanation of sleep's purpose. Knowing which dial sets the volume does not explain what the music means. Consider a sleeping body in the small hours of the night, far from morning. The limbs are still, the breath is slow, the heart has slowed slightly, the blood pressure dropped, the core temperature settled into its nightly trough. On the surface, this looks like the body's systems winding down. Inside, a different picture is forming. In the blood, a population of immune cells called T cells is more active than it was during the previous afternoon. Their surfaces are studded with adhesion molecules called integrins, and during the slow wave phases of sleep, these integrins are more fully activated than at any other time of day. An integrin activation is not a small event in an immune cell's life. It is the mechanism by which the cell grabs hold of an infected or aberrant target and begins the process of destruction or tagging for destruction. The activation is higher during sleep. The immune cell, in other words, is better at its work in the dark hours of deep sleep than during the alert hours of midday. At the same time, the pituitary gland is releasing growth hormone in a pulse that is closely tied to the onset of slow-wave sleep, a pulse so reliably timed that researchers can predict its arrival from the EEG alone. Growth hormone does not only govern growth in developing animals, it drives protein synthesis, tissue repair, and cellular maintenance across the whole body throughout life. The hours of deep sleep are the hours in which the body rebuilds most actively. The gut, the muscle, the skin, all of them carry out their fastest repair in the window when the body is still and the hormonal environment tilts toward reconstruction. This biological environment, the elevated integrins, the growth hormone pulse, the warmth of slow metabolic rebuilding, cannot be replicated in wakefulness. The hormones of waking hours, cortisol prominent among them, suppress the immune remodeling and cellular reconstruction that sleep enables. The two states are not simply active and inactive, they are specialized. Each enables things the other does not. What remains genuinely open is whether the immune enhancement and the cellular repair are the reason sleep evolved, or whether they are capabilities that evolution recruited into an already existing sleep state that originated for some other reason, and whether those two framings are even meaningfully different from each other. An animal with no brain is resting on the seafloor. It is the upside-down jellyfish, named for the habit of sitting on the substrate with its bell inverted and its tentacles trailing upward into the water column. During the hours of the night, its pulsation rate drops by a quarter or more. Its response to a disturbance, a gentle touch, a brief light, is measurably slower than it is during the daytime. If it is kept awake through the night by repeated stimulation, it pulses more slowly the following day, as if recovering. All of this is the behavioral signature of sleep. All of it is present in an animal whose nervous system is not a brain at all, but a diffuse net of neurons spread through the body's tissues, lacking any centralized processing structure, incapable of producing the oscillatory waves that the sleeping mammalian brain generates, and yet exhibiting the same four hallmarks: quiescence, elevated arousal threshold, rapid reversibility, and homeostatic rebound that define sleep wherever it is found. The jellyfish's lineage diverged from the vertebrate lineage more than 500 million years ago in a sea that would not be recognizable as any present ocean, populated by animals no museum has reconstructed. Whatever sleep is, whatever its deepest function, the jellyfish finds it necessary. An animal that has no neurons firing in sequences, no hippocampus replaying the spatial geometry of its day, no glymphatic fluid washing through a cortex, no immune T cells activated by slow-wave oscillations. This animal still needs to stop, to lower its response to the world, to spend time in a state from which homeostatic rebound is possible. The mechanisms available to it are not the mechanisms of the mammalian brain, but the function, something restored, something recovered, something that cannot wait until morning, appears to be the same. This is where the question deepens past the point where any current hypothesis reaches comfortably. If sleep in the jellyfish serves a purpose, and sleep in the mammal serves a purpose, and those purposes are served by entirely different biological mechanisms, then either sleep evolved separately in many lineages for many different reasons, or there is something so fundamental about the need for periodic rest and restoration that it emerges in any sufficiently complex living system, almost regardless of how that system is built. The first possibility would mean that what is called sleep is not one thing, but many things that happen to look similar from the outside, a convergence of behavior without a convergence of mechanism. The second possibility would mean that sleep is a near universal requirement of biological complexity itself, that building a system capable of sustained activity necessarily builds in the requirement for periodic recovery, and that the particular mechanisms of that recovery are secondary elaborations on a more basic biological imperative. Neither possibility closes the question, both deepen it. Across the ceiling of geological time, from the early Cambrian to now, some version of sleep has been present in every lineage that has been carefully studied. Not every species. There are gaps in the record, animals that have not yet been observed closely enough, organisms whose behavioral states remain uncharacterized. But among the lineages that have been examined, mammals, birds, fish, reptiles, insects, mollusks, crustaceans, cnidarians, the behavioral signatures of sleep appear reliably. The more carefully researchers look, and the more ancient the animals they study, the further back the origin of sleep recedes. There is no confirmed lineage that has fully abandoned sleep under the pressure of predation or any other selective force. Some lineages have modified it deeply, sleeping with half a brain, sleeping in fragments of seconds, sleeping during flight, but none has let it go entirely. This absence is its own kind of evidence. Evolutionary selection is not gentle with costly behaviors. An animal that spent 8 hours a day, unconscious and vulnerable, when its competitors spent those hours feeding or reproducing or monitoring their environment, would be at a significant disadvantage. If sleep could be abandoned without cost, or if the functions it serves could be performed just as well in wakefulness, then across the vastness of geological time, in some lineage somewhere under sufficient predation pressure, it would have been abandoned. That there is no confirmed case of this in any studied animal across hundreds of millions of years implies that the cost of abandoning sleep entirely exceeds whatever benefit the extra waking hours would provide. The argument is not proof. It is an inference from absence, and absence is always a soft kind of evidence. But the inference is consistent with everything else known about sleep. The lethality of its deprivation in laboratory animals, the universality of its molecular machinery, the depth of its conservation across the tree of life. Together, these threads point in the same direction. Sleep is not optional. Something in what it does is necessary. What that something is, precisely, is still being worked out. The question has been asked carefully for more than a century. It has been asked with electrodes and with imaging machines, with genetic tools and with behavioral tests, with electron microscopes that can resolve the size of a single synapse before and after a night of sleep. The answers have multiplied. The evidence for each of the major hypotheses, synaptic recalibration, memory consolidation, metabolic clearance, immune support, cellular repair, is real and growing. What has not arrived is a single framework that unifies them, or a single experiment that settles the question of which function, if any, is primary. Some researchers have proposed that asking for a single function is the wrong question, that sleep, like wakefulness, does many things simultaneously, and that the search for the function of sleep is like asking for the function of wakefulness, a question so broad that it cannot have a single answer. The brain during waking hours learns, navigates, socializes, plans, maintains its own physiology, processes sensory information, and regulates the body's organ systems. Sleep may be no different in its complexity. The hours of sleep may be hours in which the brain is doing a parallel set of essential things, each real, each necessary, each incomplete, without the others. In a narrow passage between two cells, in the sleeping cortex of an animal still hours from morning, cerebrospinal fluid moves slowly through the dark. It carries with it the dissolved residue of the day, fragments of protein, the byproducts of electrical signaling, the molecular evidence of everything the brain did between first light and last light. The fluid's movement is not fast. It is the pace of something ancient and unhurried, driven by the pulse of blood in the vessels beside it, and by the slow wave of the sleeping brain's own oscillations. In this passage, in this dark, something is being cleaned that cannot be cleaned any other way. Something is being restored that cannot be restored in waking. Whatever name is put to that process, whether it is called clearance or consolidation or recalibration or repair, the body treats it as necessary enough to spend a third of a life in its service. A third of a life. This is the arithmetic of sleep, laid out plainly. An animal that lives 30 years spends 10 of them asleep. An animal that lives 80 years spends more than 25 years in states of reduced consciousness, vulnerability, and temporary withdrawal from the waking world. Evolution has preserved this investment in every studied lineage, maintained the molecular machinery that enables it across 600 million years of separate evolutionary paths, built the circadian clock into every cell and the homeostatic pressure into the chemistry of the brain, and left no confirmed example of a lineage that found its way around it. Whatever sleep is for, its cost has never outweighed its value. Whatever it is doing in those long, still hours, it is doing something the body cannot do without. The molecular clock in the liver cell completes another cycle as the night moves on. The clock in BML1 proteins, having opened the genes and driven the accumulation of their own suppressors, find the complex gently silenced, the feedback loop arriving at its daily pause. In a few hours, the suppressors will be cleared, the complex will reform, and the cycle will begin again, the same sequence, in the same cell, as it has run every day of the animal's life, as it has run in one form or another in living cells for longer than animals have existed. The clock does not know what it is counting. It does not know that the animal is asleep, or that the animal exists, or that somewhere outside the soft walls of the cell, the night is slowly becoming morning. It simply turns, in the dark, marking time with a patience that has no beginning the historical record can reach. Sleep continues in every sleeping body, in every sleeping city, in every corner of the living world where night has fallen. The dolphin drifts through dark water with one hemisphere resting and one hemisphere alert. The migratory bird, somewhere above a dark continent, takes brief moments of unihemispheric rest between long beats of its wings.